We have been discussing one method of creating human insulin for diabetic patients. To this point, our efforts have been focused on creating a recombinant DNA plasmid that will be able to express human insulin. But, if you recall, our ultimate plan is to get bacterial cells to make insulin. That means we need to get our human insulin plasmid into a bacterial cell. Is that even feasible? Well, lucky for us, there are a number of ways to genetically modify bacterial cells. One of the most common procedures used in laboratories and classrooms alike is known as transformation. Transformation is the process in which the genetic makeup of a cell is changed by the introduction of DNA from the surrounding environment. Scientists commonly use the bacteria found in our gut in bacterial transformation experiments. This bacteria is known as Escherichia coli, or E. coli for short. E. coli is one of the most commonly used organisms in scientific experiments. Bacterial cells that are actively growing are most amenable to bacterial transformation. However, they must undergo a series of treatments before they are competent for transformation. There are several ways to make cells competent for transformation, but we will consider the simplest version, which only requires calcium chloride, ice, and heat. Recall that a cell is surrounded by a cell membrane composed of phospholipid molecules. The phospholipids are organized into a lipid bilayer in which the hydrophobic tails point inward and the hydrophilic heads point outward. Since the hydrophilic head possesses an electronegative phosphate group, the outside of the bacterial membrane is negatively charged. We have also learned that DNA is charged. The phosphate group in DNA molecules gives DNA a negative charge as well. Does anyone see a problem with this? We want to push the DNA into the cell, right? But if DNA and the cell membrane have the same charge, this is going to be like trying to push the negative side of two magnets together. The DNA and cell membrane are going to repel each other. Bathing the bacterial cells in the salt solution calcium chloride solves this problem. The calcium ions are attracted to both the negatively charged cell membrane and DNA molecules in the solution. The close proximity of the positively charged ions to these negative charges effectively neutralizes the charge on both the cell membrane and the DNA molecules. This enables the DNA plasmid to move toward the cell membrane without being repelled. Well, getting the DNA plasmid near the cell membrane is just half the story. Somehow, we need to move the plasmid into the cell. Scientists determined that a sudden increase in temperature could propel a plasmid into a bacterial cell. This treatment is called a heat shock. While the cells and DNA are incubated with the calcium chloride solution, the entire solution is chilled on ice. Then the cell DNA solution is removed from the ice bath and briefly heated to 42 degrees Celsius. This heat pulse induces some plasmids to enter bacterial cells. It is important to note that chilling the cell DNA solution is crucial for transformation to occur, because the difference between room temperature and 42 degrees is insufficient. Scientists aren't exactly sure how transformation works. One theory is that the heat shock treatment effectively opens pores in the cell membrane through which a plasmid molecule could travel. It's also believed that the heat differential between the outside of the cell and the inside of the cell may provide the force that pushes the plasmid into the cell. A plasmid is carried along as the warm water rushes through the pore